Welcome to my video presentation. The topic that I will be talking about is chlorosis. Now let me move towards my introduction. My name is Zuba Noor and I am a student of the Department of BS Biochemistry. Let me introduce you to the contents of this presentation. First, I will start with the introduction. Then I will move towards the changes that occur in plant body due to chlorosis, and then I would be talking about the factors, common causes, nutrient deficiencies, as well as the environmental factors that lead to this condition. Then I would be talking about the ways in which we can spot chlorosis as well as the importance of a proper diagnosis. After that, I would be talking about the kinds of vegetation that gets affected by chlorosis and the effect it has on plants. Then I would move towards the management strategies of chlorosis, and I would also give an example of a study that investigated the effect of chlorosis severity in a specific population of plant. In the end, I would give a conclusion. Let me start with the introduction. Chlorosis is a condition which is characterized by the yellowing or withering of plant leaves due to the lack of chlorophyll, and it is often caused by nutrient deficiencies or different kinds of diseases. Chlorophyll is basically a pigment which is responsible for the green color of plants, and it is very essential for the process of photosynthesis. In photosynthesis, sunlight is captured and it is converted into chemical energy, which is the food for the plant. This word originates from a Greek word which is chloros. It is basically a color which refers to pale green. This is because of the characteristic color change that occur in the leaves due to chlorosis. Here is a picture that shows chlorosis in raspberry leaves, and the other one shows chlorosis in jasmine leaves. In the brackets, their scientific names are given. Now I will move towards a very important point, that is chlorosis is actually a symptom and it is not a disease. Now let me talk about how chlorosis actually progresses in a plant body. In the first step, the factors that lead to this symptom disrupt the normal uptake and transport in the plant body. In the second step, chlorophyll production is decreased in the plant body, and then in the third step, the affected leaves start to lose their vibrant green color, which means that chlorosis is actually starting its onset. And then after that, in the fourth step, the plant's ability to produce chemical energy from sunlight energy becomes abnormal or impaired. Then in the last step, the condition starts progressing and becoming more and more severe if it is not addressed properly. Now I will be talking about the significance of chlorosis. Chlorosis is very significant for plant because it serves as an indicator for the underlying issues that the plant might be having, which is very harmful for its health and vitality. This means that chlorosis lets us know that the plant is having abnormalities underneath and that it is not having a healthy metabolism. The yellowing or withering of the leaves tells us that there is a disruption in the normal functioning of chlorophyll, and which again is the pigment which is responsible for the photosynthesis. By identifying chlorosis, gardeners, farmers and botanists can actually diagnose and address the specific condition which is leading to this symptom. Resolving this is very important and very helpful as photosynthesis becomes effective and the healthy growth starts to happen, and the crop yield is also maximized. Now I will move towards the changes that occur in plant body due to chlorosis. The first one is chlorosis synthesis and disruption. Chlorophyll is a pigment which is responsible for capturing light energy during photosynthesis, and it enables plants to convert sunlight energy into chemical energy. The process of chlorophyll synthesis starts in chloroplasts, which are organelles and the sites of photosynthesis, and a series of enzymatic reactions happen there. The key steps which are involved in the incorporation of the basic components include porphyrin, magnesium, amino acids, and chlorophyll molecules. Many nutrients have a very important role in the synthesis of chlorophyll, and these include nitrogen, magnesium, iron, and some others. Nitrogen is the basic key component of chlorophyll and it forms the central porphyrin ring structure. Here is a picture of the central porphyrin ring structure of chlorophyll, and it is basically a cyclic arrangement of atoms that consists of 4 pyrrole rings, and there are methane bridges that connect these units together, and it is often found at the core of molecules like chlorophyll and heme. The next important element is magnesium. It is required for the insertion of central magnesium ion into the porphyrin ring, which stabilizes the chlorophyll molecule. Iron is involved in the synthesis of the heme group, which is essential for chlorophyll formation in some plants. The name heme exactly shows us that it has something to do with iron. Here is a picture of a chlorophyll molecule, and as we can see, it consists of a porphyrin ring which is coordinated with the central magnesium ion, and there are hydrophobic side chains, and there is a phytol tail. It enables it to capture light energy during photosynthesis. Now I will be talking about how disruption of chlorophyll synthesis occur due to the deficiency in nutrients. Nutrient deficiencies can actually disrupt the synthesis of chlorophyll and it leads to chlorosis. First, let me talk about nitrogen. Nitrogen deficiency can limit the availability of the key component for chlorophyll synthesis, and it results in reduced chlorophyll content and the leaves start to become pale. Magnesium deficiency. Without enough magnesium, the insertion of central magnesium ion into the porphyrin ring is impaired, and it leads to decreased chlorophyll production and the development of chlorotic leaves starts to occur. Iron deficiency. In plants that rely on iron for heme group synthesis, iron deficiency can hinder the formation of chlorophyll and it can cause chlorosis. Next, I will be talking about the consequences of disrupted chlorophyll synthesis. Disruption of chlorophyll synthesis impairs the plant's ability to carry out photosynthesis efficiently. When chlorophyll content is reduced, it leads to a decreased capacity to capture light energy and convert it into chemical energy, which means that less light is harnessed, and this results in reduced plant growth, stunted development, and reduced overall vigor, which refers to the overall health of the plant. The next one is role of iron, magnesium, nitrogen in plant metabolism. First, I will be talking about the role of iron. Iron is a very important micronutrient which is involved in many aspects of plant metabolism, including the production of chlorophyll. Iron is a key component of the heme group, which is necessary for the synthesis of chlorophyll in many kinds of plants. Iron is also a cofactor for enzymes which are involved in various metabolic processes, which include respiration, nitrogen fixation, and DNA synthesis. Electron transfer reactions. Iron is also involved in the electron transfer reactions which happen in the electron transport chain during the process of photosynthesis and respiration, and it plays a very important role in the production of energy in the plant body. The next element is magnesium. Magnesium, as we know, is a central element in chlorophyll molecules, and thus it makes it a very essential molecule for the production of chlorophyll. The role of magnesium in the synthesis of chlorophyll is that it binds to the porphyrin ring structure, and it makes the chlorophyll molecule stable and enables its light capturing properties. It is also a cofactor for many enzymes which are involved in carbohydrate metabolism, protein synthesis, and energy transfer processes. It is also required for the proper functioning of photosystems, electron transport, and ATP synthesis during the process of photosynthesis. The next element is nitrogen. Nitrogen is a very critical macronutrient which is required for plant growth, chlorophyll synthesis, and overall metabolic processes occurring in the plant body. The role of nitrogen in chlorophyll synthesis is that it is a very important component in chlorophyll molecule, and it forms the central porphyrin ring structure. It is also very crucial for the synthesis of amino acids, which are the building blocks of proteins, and they are involved in various metabolic pathways and enzyme activities. It is also required for the production of nucleotides, which are essential for DNA and RNA. DNA and RNA are nucleic acids, and these enable the proper genetic expression and cellular processes of the plant. Third kind of change that happen are the physiological effects. The first one is reduced photosynthesis. Nutrient deficiencies can lead to a decrease in the production of chlorophyll, which is the pigment responsible for capturing light energy during photosynthesis. This reduction results in a diminished photosynthetic capacity, which therefore as a result causes a decrease in the energy production, and the carbohydrate synthesis is also limited as a result. The second one is impaired nutrient transport. Nutrient deficiencies can disrupt the transport of essential elements within the plant. For example, the deficiencies in macronutrients like nitrogen and potassium can hinder their movement from older leaves to younger tissues, which affects the overall nutrient distribution and allocation within the plant. The third one is altered enzyme activity. Nutrient deficiencies can impact enzyme activity as well. Many enzymes require specific nutrients as their cofactors for proper functioning. When a cofactor binds with an enzyme, the enzyme starts its proper functioning. Insufficient nutrient availability can lead to reduced enzyme activity and even make it inactive, which affects various metabolic processes and also affects the overall health of the plant. The fourth one is disrupted hormonal regulation. Nutrient deficiencies can also disrupt the hormonal balance within the plant. For example, the deficiencies in micronutrients like iron or zinc can interfere with the synthesis or the signaling of plant hormones, which play a very important role in growth, development, and stress response. The fifth one is impaired protein synthesis. Nutrient deficiencies, especially the ones which involve essential amino acids, can lead to a reduced protein synthesis. Proteins are very essential for many cellular processes, including enzymatic reactions, structural support, and signal transduction. When they are not sufficient, it can negatively impact the plant physiology. The sixth one is increased susceptibility to stress. Nutrient deficiencies can make the plant weaker and also more vulnerable to biotic and abiotic stressors. Biotic stressors are the ones which are the living components, and abiotic stressors are the ones which are non-living components of the environment. Without enough nutrients, plants will also have a compromised defense mechanisms and a reduced stress tolerance, increased susceptibility to pests, diseases, and outer environmental stresses. Now I'll be talking about the factors that lead to chlorosis, and these include nutrient deficiencies, inadequate light levels, pH imbalances in the soil, diseases, environmental stresses, and poor drainage. Next, I will be talking about the causes that count towards the onset of chlorosis. The first one being nutrient deficiencies, the second one is soil conditions, and the third one is pathogens and diseases. Let me first talk about nutrient deficiencies. Nutrient deficiencies lead to chlorosis in a number of ways through a number of mechanisms. First one is inadequate availability of nutrients. When essential nutrients like iron, magnesium, nitrogen, etc. are lacking in the soil, the plants cannot have them for proper metabolic processes, and these nutrients are very important for the synthesis of chlorophyll and its proper functioning. The second one is disrupted nutrient transport. Nutrient deficiencies can disrupt the normal transport of essential elements in a plant. For example, magnesium and iron deficiencies hinders the transport from roots to leaves, and therefore it affects the production of chlorophyll. The third one is impaired enzyme activity. Many essential nutrients act as cofactors for enzymes, as we talked about earlier. Most of these enzymes are involved in the synthesis of chlorophyll and different kinds of metabolic reactions. These cofactors, as I explained earlier as well, are non-protein molecules or ions that bind to enzymes. Deficiency in these nutrients can reduce the enzyme activity, and it therefore leads to an insufficient production of chlorophyll. The fourth one is disrupted electron transport chain. Nutrients like iron are necessary for electron transplant in chloroplast, which is essential for the conversion of light energy into chemical energy. A deficiency in these nutrients can disrupt the chain, and it affects the photosynthesis and chlorophyll formation. The fifth and last one is altered pH and nutrient availability. Nutrient deficiencies can be made worse when they are paired with imbalance in the soil pH. For example, different nutrients become less available to plants in alkaline or acidic soils, which hinders their absorption and utilization by the plant, and therefore it contributes to chlorosis. The second one is soil conditions. Soil conditions can also contribute to chlorosis in many ways. The first one is pH imbalances. High or low soil pH can affect the nutrient availability for plants. For example, alkaline soil which have high pH can lead to iron or magnesium deficiencies, whereas acidic soils which we know have low pH can result in calcium or magnesium deficiencies, and both of these can trigger chlorosis. The second one is poor soil drainage. Waterlogged or poorly drained soil can create an oxygen-deprived condition that can hinder the root function and the nutrient uptake of the plant, and this can result in chlorosis because of the reduced availability and absorption of the important nutrients required by the plant. The third one is nutrient imbalances. Soil nutrient imbalances, for example, excessive levels of certain elements or imbalanced ratio between different nutrients can interfere with nutrient uptake by the plant and it leads to chlorosis. Soil compaction. Compacted soils are the soils that have very closely packed particles and there are very less air spaces between them. The fifth one is soil texture and structure. Soil texture, for example, heavy clay soils or sandy soils can influence the nutrient retention and availability. Inadequate or insufficient nutrient retention or excessive draining can contribute to nutrient deficiencies which therefore result in chlorosis. Clay soils are the type of soils that have high water retention and water cannot easily percolate through it, whereas sandy soils are the type of soils that have very poor water retention and water can easily percolate through it. The third one is pathogens and diseases. So pathogens are harmful living organisms that cause different kinds of diseases. These can contribute to chlorosis in plants through different mechanisms. The first one being direct damage to chlorophyll. Different kinds of pathogens can directly infect and damage chloroplasts where the chlorophyll is located, and this damage can disrupt the normal chlorophyll synthesis or even degrade existing chlorophyll molecules. The second one is nutrient deprivation. Pathogens can interfere with the nutrient uptake or utilization by plants. They may compete with plants for essential nutrients, disrupt the nutrient transport systems, or induce nutrient imbalances, all of which can contribute to nutrient deficiencies and therefore as a result lead to chlorosis. The third one is toxin production. Some pathogens produce toxins that directly affect chloroplasts or interfere with chlorophyll synthesis, leading to chlorosis. These toxins can inhibit enzymes involved in chlorophyll production or damage chloroplast membranes, which can therefore impair photosynthesis. The fourth one is vascular system disruption. The vascular system is the system that mainly includes the stem and the transport through it. Certain pathogens can invade and colonize the plant's vascular system, which as a result blocks and impairs the transport of water, nutrients, and sugars through the plant. This disruption can result in deficiency of certain nutrients, including the ones that are important for chlorosis, and then they can result in chlorosis. The fifth and last one is induced stress response. Pathogen infections often trigger a stress response in plants, which results in altered physiological and metabolic processes. And this stress response can lead to chlorosis and, as a secondary effect of plant, redirecting resources away from chlorophyll synthesis to the defense mechanisms against the pathogen. Now I will be talking about the nutrient deficiencies which lead to chlorosis. The first one being iron, then magnesium, nitrogen, zinc, and manganese. First, let me talk about iron chlorosis. Now, let me start with an introduction. Iron chlorosis is a plant disorder which is characterized by the yellowing or withering of plant leaves due to insufficient iron availability. It is a common nutritional disorder and it affects various plant species including trees, shrubs, and crops. Iron is an essential micronutrient which is required for chlorophyll synthesis and many other important processes in the plant body. Now I will be talking about the causes and symptoms of iron chlorosis. Soil factors. Iron chlorosis often occurs in alkaline soils where the high pH levels limit the availability and uptake of iron by the roots, and in such conditions iron becomes chemically unavailable or it may exist in such a form that may not be easily absorbable by the plant. The second one is poor iron mobility. Iron has limited mobility in plants, which means that it cannot easily move from older leaves to younger leaves. Therefore, iron deficiencies primarily affect the new or young leaves, which lead to chlorosis symptoms in those tissues. The symptoms of iron chlorosis include the interveinal yellowing of the leaves, where the veins remain green, whereas the spaces between them turn yellow or white. In severe cases, the leaves may become completely white and exhibit necrosis. Now I will be talking about the impact and consequences of iron chlorosis. The first one is reduced photosynthesis. Iron, as we know, is very essential for the synthesis of chlorophyll, and its deficiency holds the plant's ability to carry out photosynthesis effectively. When photosynthetic activity is impaired, the energy production and overall growth is also affected. Second one is stunt growth and yield loss. Iron chlorosis can lead to stunted growth, decreased plant vigor, and lower crop yields. And Insufficient iron availability negatively affects the plant metabolism, nutrient transport, and reproductive processes. The third one is increased susceptibility to stress and disease. Iron deficient plants are more susceptible to various stresses, including temperature, drought, and disease. Weak plants are more prone to infections, and they also struggle to recover from different kinds of external or internal conditions. Now I'll be talking about the management and treatment of iron chlorosis. The first one is soil modification. Soil changes such as sulfur or acidifying agents can lower the soil pH, which can make iron more available for plant uptake. However, it is often a long-term approach, and it can also require a long ongoing maintenance. The second one is foliar applications. Foliar sprays, which contain chelate iron compounds, can be used to provide an immediate iron supply to the leaves. These sprays allow for direct absorption of iron by the foliage. The third one is soil and root applications. Applying iron fertilizers or iron chelates directly to the soil or near the root areas can provide a long-term supply of iron to the plant. The fourth one is cultivar selection. Choosing iron-efficient plant varieties or species that are naturally adapted to high pH soils can lessen the risk of chlorosis. Here is a picture of soybean leaves which shows chlorosis due to the deficiency of iron. The second one is magnesium chlorosis. Let me start with an introduction. Magnesium chlorosis is a condition which is characterized by the yellowing or whitening of plant leaves due to insufficient availability of magnesium. Magnesium is an essential macronutrient which is required for many plant processes including chlorophyll formation, enzyme activation, and energy production. Now let me talk about the causes and symptoms of magnesium chlorosis. The first one being soil factors. Magnesium chlorosis usually occurs in sandy or acidic soils which naturally have a low magnesium content. These soils also have poor water retention, and they can also have limited magnesium availability for the plant to absorb. The next one is nutrient imbalances. Imbalances in soil nutrients, especially high levels of potassium or calcium, can interfere with magnesium uptake. Excessive potassium or calcium competes with magnesium, and therefore it leads to magnesium deficiencies. Now let me talk about the symptoms. The primary symptom of magnesium chlorosis is the yellowing of leaves, which start from the leaf margins and then it slowly progresses towards the center. The veins often remain green, and this intraveinal chlorosis is a very clear characteristic of magnesium deficiency. Now I will be talking about the impact and consequences of magnesium chlorosis. The first one being impaired photosynthesis. Magnesium is crucial for chlorophyll synthesis, and its deficiency can affect the plant's ability to capture sunlight and convert it into chemical energy. Reduced chlorophyll levels can lead to decreased photosynthetic efficiency and overall plant productivity. The second one is altered nutrient uptake. Magnesium has a very important role in nutrient transport within the plants, and its deficiency can disrupt the absorption and translocation of other essential nutrients. And it can then lead to secondary deficiencies and impact plant growth and development. The third one is weakened plant health. Magnesium deficient plants are more susceptible or prone to environmental stresses, diseases, and pest attacks. Now I will be talking about the management and treatment of magnesium chlorosis. The first one being soil amendments. Applying magnesium containing fertilizers or amendments such as dolomitic lime or magnesium sulfate, which is also called Epsom salt, can increase the magnesium content in soil. Next one is foliar sprays. Foliar application of magnesium fertilizers can provide a quick and direct supply of magnesium to the plant, and it is particularly very useful for addressing acute deficiencies or when the soil uptake is limited. The third one is pH management. Maintaining the appropriate soil pH levels is crucial for magnesium availability. Adjusting soil pH to the optimal range for magnesium uptake, which is actually about 6 to 7 on the pH scale, and it can therefore enhance the magnesium absorption by plant roots. The fourth and last one is crop rotation and organic matter. Incorporating organic matter, for example, organic fertilizers in soil, can help improve the soil fertility, nutrient availability, and also reduce the risk of magnesium chlorosis. Here is a picture of tomato leaves showing chlorosis due to the deficiency of magnesium. Now I will be talking about nitrogen chlorosis. First, let me start with an introduction. Nitrogen chlorosis is a condition characterized by the overall yellowing or pale green coloration of plant leaves due to insufficient nitrogen availability. Nitrogen is a very important macronutrient which is essential for plant growth, protein synthesis, and overall metabolic processes. Now let me talk about the causes and symptoms of nitrogen chlorosis. The first one being nitrogen deficiency. Nitrogen chlorosis of plants do not receive an adequate or enough supply of nitrogen from the soil or other nutrient sources. It can be a result of low nitrogen content in the soil or imbalances in nutrient availability. The second one is high nitrogen demand. Plants which have a high nitrogen demands, for example, those which are in fast or rapid growth phases or which are heavy nitrogen consumers can experience nitrogen chlorosis if their nutrient needs exceed the available supply. The primary symptoms of nitrogen chlorosis is the overall yellowing or light green coloration of the plant tissues, which affect both the older and younger leaves. Unlike other forms of chlorosis, nitrogen chlorosis tends to affect the entire leaf rather than showing interveinal yellowing pattern. Now I will be talking about the impact and consequences of nitrogen chlorosis. The first one is reduced growth and vigor. Nitrogen is essential for promoting vegetative growth and overall plant vigor. Nitrogen chlorosis can also lead to stunted growth, delayed development, and smaller plant size. The third one is impaired photosynthesis. Nitrogen has a very important role in chlorophyll synthesis, and when it is deficient, it can affect the plant's ability to carry out photosynthesis effectively. Reduced chlorophyll levels can then hinder the plant's capacity to capture and utilize light energy for its growth and productivity. The third one is decreased nutrient balance. Nitrogen deficiency can disrupt the balance of other essential nutrients as well, and it can then result in imbalances in nutrient uptake and utilization, which can then lead to secondary nutrient deficiencies and further impact plant health and overall vitality. Next, I will be talking about management and treatment of nitrogen chlorosis. The first one being nitrogen fertilization. Applying nitrogen-rich fertilizers can replenish nitrogen levels in the soil, and it can provide the necessary nutrient supply for the plants. The choice of nitrogen fertilizer type and application timing should also consider the specific crop requirement and the soil conditions. The second one is organic matter and cover crops. Incorporating organic matter into the soil and utilizing nitrogen fixing cover crops can contribute to the long-term availability of nitrogen, and it can improve soil fertility. The third one is crop rotation and proper nutrient management. First, let me explain what crop rotation is. Crop rotation means to grow different crops in a specific sequence on the same area of land over time, expecting to improve soil health, to manage pests, and to increase the yield production. Implementing this practice and maintaining the proper nutrient management strategies can help optimize nitrogen utilization, minimize the loss, and ensure an adequate supply for plants. The fourth one is soil testing and monitoring. Regular soil testing can assess nitrogen levels and guide appropriate fertilizer application. Monitoring the plant health and observing the visual symptoms of nitrogen deficiency can also help identify and address issues in a timely manner. Here is a picture of lemon leaves which are showing chlorosis due to the deficiency of nitrogen. Now I will move towards zinc chlorosis. I will first start with an introduction to zinc chlorosis. Zinc chlorosis is usually caused by an insufficient availability of zinc. Zinc is a very important micronutrient which is required for various plant processes including enzyme activation, protein synthesis, and growth regulation. Next, I will be talking about the causes and symptoms. The first one being zinc deficiency. Zinc chlorosis occurs when plants do not receive an adequate supply of zinc from the soil or other sources. It can also be a result of low zinc content in the soil or conditions that can limit the uptake of zinc and its availability to plant roots. The second one is high pH, and zinc availability is reduced when the soil is alkaline, in which the pH is very high, and these soils often bind to zinc and make it less accessible for the plant to uptake, and it can lead to zinc chlorosis. The symptoms include mainly the interveinal yellowing of the leaves, where the spaces turn yellow or white while the veins remain green. In severe cases, the leaf tissue can also become necrotic or show different kinds of deformities. Necrosis is the process in which the tissues become dead. Next, I will move towards the impact and consequences of zinc chlorosis. The first one is reduced growth and development. Zinc is involved in various growth processes including cell division and cell elongation, and the chlorosis due to zinc can lead to stunted growth, delayed flowering, and overall reduced plant development. The second one is impaired nutrient uptake and utilization. Zinc deficiency can interfere with the uptake and transport of essential nutrients within the plant, and this can lead to imbalances and deficiencies of other minerals, and it can further affect plant health and productivity. The third one is decreased disease resistance. Zinc plays a very important role in plant defense mechanisms, and it can influence disease resistance and stress tolerance. Zinc deficient plants are usually more susceptible to infections and environmental stresses, and this compromises their overall health and well-being. Next, I will move towards the management and treatment of zinc chlorosis. The first one being soil amendments. Adding zinc-containing fertilizers or amendments to the soil can increase the available zinc for plant uptake, and common zinc fertilizers include zinc sulfate or chelated zinc compounds. The second method is foliar sprays. Foliar sprays or zinc fertilizers can provide a direct and quick supply of zinc to the plant, and this method is especially very useful for addressing acute deficiencies or when the soil uptake is limited. The next one is pH management. Adjusting soil pH to the optimal range for zinc availability, which is actually around 6 to 7 on the pH scale, can improve zinc uptake by the plant roots. Soil amendments or acidifying agents may also be necessary to achieve the desired pH range. The next one is crop rotation and organic matter. Practicing crop rotation and incorporating organic matter into the soil can improve soil fertility and nutrient availability, which includes zinc. Here is a picture of a maple tree in which the leaves are showing chlorosis due to the deficiency of zinc. Now I will be explaining what happens in the case of a nutrient overload. The nutrients being iron, magnesium, nitrogen, zinc, and manganese. First, I am talking about iron. When it is overloaded in a plant, it can lead to oxidative stress and toxicity in the plant, and it can hinder the nutrient uptake and cause the leaves to burn. In the case of magnesium excess, the plant can disrupt the balance of other nutrients, and it can lead to nutrient imbalances and impair the growth and development of the plant. In the case of nitrogen, it can promote excess vegetative growth and delay flowering and fruiting, and increase susceptibility to diseases and make it more prone to pathogens. In the case of zinc, when a plant is overloaded, it can inhibit or stop the uptake and utilization of other essential nutrients and can lead to nutrient imbalances and impair plant growth and development. Next, I will be talking about the environmental factors which lead to chlorosis. These include temperature, light intensity, water availability, and air pollution. First, I will be talking about temperature. In case of extremely high temperature, the nutrient uptake and utilization is affected, and it can then lead to chlorosis. When transpiration is increased due to high temperatures, the soil is deprived of the moisture contents, and it can hinder the nutrient movement towards the roots. The next one is impaired nutrient absorption. Heat stress can disrupt the function of the roots, and it can reduce the nutrient absorption and transport. Then the next one is altered enzyme activity. Extreme heat denatures the enzymes which are involved in nutrient metabolism and can affect the utilization of the enzyme. Denaturation is the process in which a molecule breaks down into smaller components. The next one is increased nutrient demand. High temperatures can boost plant metabolism and can increase nutrient requirements and potential deficiencies. Next, I'll be talking about the impact of low temperatures on nutrient uptake and utilization. When the temperatures are extremely low, it can impact the plant in a number of ways and can lead to chlorosis. The first one being reduced nutrient mobility. In cold temperatures, the nutrient movement is limited within the plant, and it affects the nutrient distribution to developing leaves. The next one is slowed metabolic processes. When the temperature is low, it can hinder the nutrient absorption and can reduce the nutrient metabolism. The next one is limited root activity. In cold temperatures, root growth is inhibited and it decreases the nutrient uptake from the soil. The fourth one is increased solubility issues. In cold temperatures, solubility issues are caused in the soil, which affects the availability of a nutrient. The next one is light. First, I will be talking about inadequate light. When the light is not sufficient, it can disrupt photosynthesis and chlorophyll production and can lead to chlorosis. The first effect is reduced energy capture. When light is not enough, the ability of chlorophyll to capture light energy is reduced and the carbohydrate production also goes down. So does the chlorophyll synthesis. The next one is decreased electron flow. Is when the light intensity is low, the electron flow is slowed down and it impacts the ATP and NADPH production, which is actually very necessary for the biosynthesis of chlorophyll. The next effect is stunted growth. When light is not sufficient, it causes elongated stem, sparse or very less foliage, and pale or less pigmented leaves due to the limited chlorophyll content. Now I will be talking about what happens in an excess of light. When light is in excess, it disrupts photosynthesis and chlorophyll production and can lead to chlorosis. The first one being photoinhibition. Intense light exceeds the photosynthetic capacity and can damage the apparatus and reducing chlorophyll synthesis. After that, the next one is reactive oxygen species, which is abbreviated by ROS. Excessive light generators ROS damage chlorophyll and impair photosynthesis. The next one is photobleaching. Longer exposure to excessive light can lead to chlorophyll photobleaching, which reduces light capture and then results in chlorosis. The next one is water stress. First, I'll be talking about the consequences of drought. Water stress, such as a drought condition, significantly impact nutrient uptake in plants and can lead to chlorosis. The first one is reduced root absorption. In case of a drought, the availability of water is limited in the soil, and this causes the roots to face difficulty in absorbing nutrients. And as the water becomes scarce, the movement of essential elements towards the roots is hindered or stopped, and it results in reduced nutrient uptake and availability for the plant to utilize. The next one is impaired nutrient transport. Water stress disrupts the flow of nutrients within the plant's vascular system, and without sufficient water to facilitate the nutrient transport, the movement of essential elements from older leaves to younger tissues is delayed. This leads to nutrient imbalances and chlorosis in the developing leaves. Now I'll be talking about the case of excessive irrigation. Excessive irrigation can also have negative implications for nutrient uptake, and this contributes to chlorosis. The first one being leaching of nutrients. Overwatering can lead to the leaching of nutrients from the soil, as excessive water carries away essential elements and reduces their availability for the plant uptake and utilization. It results in nutrient imbalances, and as a result, the plant starts to develop chlorotic symptoms. The next one is oxygen deprivation. Prolonged water saturation due to excessive irrigation can lead to oxygen deprivation in the root area of the plant, and insufficient oxygen levels start to impair root function and reduce the nutrient absorption efficiency, and therefore it contributes to nutrient deficiencies and chlorosis. The next one is air pollution. First, I will be talking about pollutants like ozone. Ozone, which can be an air pollutant, can have detrimental effect on plant health and can contribute to chlorosis. The first one being oxidative stress. Ozone is a reactive gas, and when it enters the leaf tissues, and it can initiate oxidative stress. It can damage the chloroplast, disrupt chlorophyll synthesis, and impair photosynthetic processes because it is very reactive. Thus, it leads to chlorotic symptoms. The next one is reduced photosynthetic efficiency. Ozone interferes with the functioning of photosystem 2, which is responsible for capturing light energy during the process of photosynthesis, and this disruption results in reduced photosynthetic efficiency, and it limits the energy production and chlorophyll synthesis, which ultimately leads to chlorosis. Now I will be talking about the next pollutant, which is sulfur dioxide. Sulfur dioxide can also contribute to chlorosis in plants in the following ways. Sulfur deficiency. High levels of sulfur dioxide in the air can inhibit the uptake and utilization of sulfur by plants and lead to sulfur deficiency. Sulfur is a very essential nutrient which is involved in synthesis of chlorophyll, and insufficient sulfur availability can result in chlorophyll degradation and can cause chlorotic symptoms as a result. The next one is oxidative damage. Sulfur dioxide can generate sulfur trioxide and sulfuric acid in plant tissues, which leads to oxidative damage. This oxidative stress affects chloroplasts and disrupts chlorophyll production and contributes to chlorosis. Now I will be talking about the additional factors which can cause chlorosis. First, viral infections. Certain plant viruses can interfere with the chlorophyll synthesis and can disrupt the cellular processes, which leads to chlorotic symptoms in the plants which are affected by the virus. The next one is root damage. Physical damage to plant roots from mechanical injury or improper transplantation can impair the nutrient uptake and transport, which can result in chlorosis. The next one is environmental stressor. Extreme temperatures, drought, waterlogging, salinity, or pollution, etc., can cause stress to plants and affect their nutrient uptake, their metabolism, and its overall health, which can lead to chlorosis. Next, I will be talking about how we can spot chlorosis. The first one being leaf color changes. Chlorosis is commonly characterized by the yellowing of leaves, and whereas the veins usually retain their green color. This condition indicates a reduction in chlorophyll production or impaired chlorophyll function. The yellowing occurs because chlorophyll, which is responsible for the green color of the leaves, is either insufficient or it breaks down, which reveals the other pigments in the leaf. The second one is leaf pattern. Chlorosis occurs in plants in two different patterns. The first one is general chlorosis and the next one is interveinal chlorosis. In general chlorosis, the entire leaf turns yellow and it indicates a deficiency of a specific essential nutrient, while in the second pattern, the areas between the leaf veins turn yellow while the veins themselves remain green, which indicates a specific nutrient deficiency which affects the chlorophyll production. Other symptoms can include reduction in leaf size, curling or distortion of leaves, and a premature leaf drop. These symptoms can further indicate different kinds of physiological stresses that the plant is experiencing due to nutrient deficiencies or other underlying factors which are causing yellowness in the leaves. Now I will be talking about the diagnostic tools. The first one being soil testing. It is a valuable tool for diagnosing. It is a valuable tool for plant diagnosis. Soil samples are analyzed to determine the nutrient levels and the pH, and comparing the results with optimal specific nutrient ranges can help identify the deficiencies or excesses. The next one is pH measurement. Soil pH plays a very important role in nutrient availability, and extreme pH levels can hinder the nutrient uptake and can cause chlorotic symptoms. Soil testing also helps determine if the pH adjustments are required in the treatment or diagnosis or not. The next one is tissue analysis. It involves testing the plant tissues in a laboratory. Nutrient concentration assessment. The first way is nutrient concentration assessment. Plant samples are collected and are analyzed for nutrient content, and comparing the results with established sufficiency ranges can help identify the nutrient deficiencies or the imbalances which are associated with the yellowing of the leaves. The second way is key nutrient ratios. Tissue analysis can also assess the ratio between nutrients, for example, nitrogen to potassium ratio, and these imbalances in the ratios can indicate specific nutrient deficiencies which are contributing to the yellowing. The next one is visual symptom identification. Observing and analyzing visual symptoms which are shown by the plants being affected by the yellowing, which are shown by the plants, can provide a valuable insight of the causes. Different nutrient deficiencies produce characteristic visual symptoms, and understanding these patterns can help pinpoint the nutrient which is most likely to be involved in the yellowing. Reference guides can also be used in identification, and these guides have visual comparisons of healthy and deficient plants in them, which can be used by farmers, gardeners, and botanists. Now I will be talking about the importance of a proper diagnosis. First of all, why is a proper diagnosis important? Proper diagnosis is very important for effectively managing onset of chlorosis in plants, because if it is not treated, it can keep on continuing. It can also be caused by viral factors including nutrient deficiencies, viral infections, root damage, or environmental stressors. Treating chlorosis without identifying the root cause can result in ineffective or temporary solutions which are not quite helpful in the long run. The next one is diagnostic process. The accurate diagnosis involves careful observation of the plant's symptoms, considering the environmental conditions, the laboratory tests, and the experts. Symptoms should be examined very thoroughly. For example, the pattern of discoloration, the age of affected leaves, and the accompanying signs of pests or diseases. Next, targeted interventions. Identifying the precise cause of chlorosis allows for targeted interventions which are tailored for the specific issue. For example, in case of nutrient deficiencies, applying appropriate fertilizers or amendments with the deficient nutrient can address nutrient deficiencies and restore the health of the plant. In case of viral infections, implementing measures such as plant removal, disease-resistant varieties, or preventive treatments can help manage chlorosis very well. In case of root damage, improving root health, proper irrigation practices, or replanting damaged roots can be necessary in the treatment. Next, environmental stressor. In case of environmental stressors, adjusting the watering schedules, providing shade, improving soil drainage, stopping pollution can help manage chlorosis, which is caused by these factors. The next one is long-term health and prevention. By accurately diagnosing the underlying cause of chlorosis, plant owners or growers can employ effective management strategies which can lead to improved plant health and recovery from chlorosis and prevent any future occurrences. Regular monitoring and addressing of any underlying issues that can contribute to chlorosis can also help maintain the long-term health and productivity of plants. Now I'll be talking about the kinds of vegetation which are affected by chlorosis. The first one being crops, second ornamental plants, and third being trees. First I will be talking about crops. Several crops are prone to chlorosis, which include soybeans, corn, citrus trees, tomatoes, spinach, and many other vegetables. These crops are particularly susceptible to nutrient deficiencies like iron, magnesium, or nitrogen, which can lead to these symptoms and hinder their growth and development. The second one being ornamental plants. These include azaleas, roses, and hydrangeas. These plants are commonly grown for their aesthetic value, and these can develop chlorotic symptoms when they are faced with nutrient imbalances. Proper nutrient management is very essential to maintain their vibrant and healthy appearance. The next one being trees. These include maples, oaks, and birches. These can develop the yellowing leaves or reduced growth or an overall decline when the essential nutrients are lacking and the needs are not properly met. Now I'll be talking about the effect of chlorosis on plants, which includes reduced photosynthesis, stunted growth, lower disease resistance, impact on production, and lower yield. Next, I will be talking about the management strategies of chlorosis. First, I will be talking about soil amendments. Appropriate fertilizers and soil supplements have a very important role in correcting nutrient deficiencies and restoring the health of a plant. The first one is balanced formulations. Fertilizers that contain specific nutrients which lack in the soil can be applied to increase the levels of specific nutrients in the plant and therefore get rid of chlorosis. The next one is slow release and controlled release fertilizers. These formulations provide a slow release of nutrients over time and ensure a consistent supply for plant uptake. The third one is organic amendments. Compost, manure, or other organic materials are very helpful in improving soil fertility, and they enhance nutrient availability and also promote microbial activity, which aids in nutrient absorption and thus it reduces the chlorotic symptom. The fourth one is pH adjustments. Soil supplements like lime or sulfur can be used to modify the pH levels during optimal nutrient availability for the plants to uptake. The next one is pH adjustment. Adjusting the soil pH is very important for enhancing nutrient availability and to get rid of chlorosis, which can be caused by alkalinity or acidity. Alkaline soils. Adding amendments like elemental sulfur or acidifying fertilizers can lower the pH and reduce the alkalinity and improve the availability of essential nutrients like iron and manganese. For acidic soils, lime and alkaline materials which raise the pH are applied, and this reduces the acidity and enhances the availability of nutrients like phosphorus, potassium, and calcium. The third one is cultural practices. Maintaining proper irrigation and avoiding over-fertilization as well as promoting good soil drainage is very important for the prevention of chlorosis. First important cultural practice is proper irrigation. Consistent and appropriate watering provides both water stress and water logging, and ensuring an optimal nutrient uptake and also prevents nutrient imbalances. The next one is avoiding over-fertilization. Excessive fertilizer application can also lead to nutrient imbalances and toxicity, and it can affect the overall health of the plant. The third one is good soil drainage. Ensuring sufficient drainage in the soil prevents waterlogged conditions. It can hinder the root function, nutrient uptake, and can lead to oxygen deprivation, and ultimately it can lead to chlorosis. The fourth one is plant selection. Selecting plant varieties which are more tolerant to specific nutrient deficiencies or environmental conditions is also a very effective strategy. The first one being nutrient tolerant varieties. Some plant varieties have a better tolerance or efficiency in the uptake of nutrients, and it reduces the likelihood of developing chlorotic symptoms. The second one is environmental adaptation. Choosing plant varieties that are well adapted to specific environmental conditions like high or low pH soils can also minimize the risk of nutrient imbalances, which results in chlorosis. Now I will be giving an example of a case study which was conducted by ResearchGate in Turkey. This study was presented on ResearchGate, and it focuses on the classification of plants in the F2 generation based on their leaf chlorosis scale. F2 means second familial generation, which is gained by the crossing over between the first familial generations. This research investigated the severity and distribution of leaf chlorosis in different plant populations and provided valuable insights into their inheritance patterns and the potential genetic factors which influence chlorosis. The results of this study contributed to understanding the underlying mechanisms and developing strategies to manage chlorosis in plants. Here is a graph which shows the data which was obtained through this research. On the horizontal axis, we have the scale for leaf chlorosis, which indicates the severity of chlorotic symptoms, and on the vertical side, we have the number of plants in the F2 generation. As we can see, different populations are showing different range of chlorotic symptoms in them, where some of them have high numbers, some of them have a low range, and the first variety has 0 values for the ranges. Mystery Bay is the city in which this research was conducted. This research is mainly showing that chlorosis is actually differently distributed in different varieties and populations of plants depending upon their susceptibility to different kinds of internal and external factors. Now I will be ending the presentation with the conclusion. To mitigate a biological problem like chlorosis, the following sustainable agricultural practices should be adopted. This can minimize nutrient imbalances, can reduce environmental impacts, and can promote long-term soil and plant health. The first one is effective nutrient management, effective water management, soil health, pest management. We can combat chlorosis efficiently by adopting these strategies and can cultivate a more sustainable and productive agricultural landscape in this way. Here is a link that shows all the references and articles that were used in making this presentation, and here is a QR code that can be scanned to access all of those articles. Thank you for viewing this video presentation. I hope that you gained some valuable knowledge from this, and also make sure to like, comment and subscribe to the channel below. Thank you.